Everyone and welcome to Unacademy. Today we are going to talk about meiosis 2. Now we have already learned about meiosis 1 and it was the process in which the number of chromosomes in the daughter cells reduced to half as that of parent. Now we are going to study meiosis 2, during which the number of chromosomes in the progeny and in the parent are just the same. That is why we call this as equational division. So if after meiosis 1 the number of chromosomes in a cell is n, then after meiosis 2 also the number will remain n only. So this is the very clear cut representation of meiosis 2, that is equational division, equal. Now this process is just similar to mitosis but always occurs in haploid cells. This is going to occur only in haploid cells, not in diploid cells. Now the main event that occurs is the separation of sister chromatids. In case of meiosis 1, separation of homologous chromosomes occurred. Here separation of sister chromatids is going to occur. Now we are going to come through different phases of meiosis 2. The first is prophase 2. Prophase 2 is a very short phase. It is not that complex as prophase 1 with other subphases also. It is just a simple process. The univalents which are present with their sister chromatids. These are the two sister chromatids and these are the two sister chromatids. And these are called as univalents. They don't form a complex now. They don't pair up. They don't form a bivalent. They are single. So they are called as univalents. So these univalents would condense. For the process of meiosis 2, they start condensing up. They condense. The nucleolus and the nuclear envelope again disappears and the chromatin material also condenses. Next is a metaphase 2, where these univalents with each univalent consists of two sister chromatids. This is a univalent. This is another univalent. These two together form a univalent and these two are the sister chromatids of this univalent. So these univalents align at the equator, this time forming a single metaphasic plate because each univalent has a single centromere. That is why a single metaphasic plate is formed. Now what is the structure that helps in attaching the spindle microtubules with the chromatids is the centromere. And what on centromere? On its surface a disc-like structure called as kinetochore is basically the reason why the chromatids attach to spindle microtubules. Next is the anaphase 1, where the shortening of the spindle microtubules occur and the centromere also splits. Now here's an important point to note. In case of anaphase 1 of meiosis 1, the centromere splitting does not happen. But in case of anaphase 2 of meiosis 2, centromere splits. And these are the two sister chromatids which have separated from each other. These are the two which have separated from each other. And the two separated sister chromatids move towards the opposite poles. Finally comes the end phase, that is telophase 2. Here the spindle fiber disintegrate, chromatids reach the poles and start uncoiling. They will again uncoil and they will become decondensed and again they form chromatin material. Nuclear envelope and nucleolus reappears. The final result is the formation of two haploid cells. Cytokinesis. Cytokinesis is division of cytoplasm. Cytokinesis is similar as in case of mitosis. In case of animal cells, the furrow is formed from outside and it grows inside in a centrifugal manner, splitting the single cell into two daughter cells. This time the daughter cells are haploid. But in case of plant cells, a cell plate is formed by the phragmoplast, which deepens from inside to outside in a centrifugal manner and finally meets the cell wall, splitting the single parent cell into two daughter cells. Again, these daughter cells would be haploid. So finally, after meiosis 2, we are getting 4 haploid daughter cells from a single diploid parent cell. This is the whole process of meiosis, and it is leading to lessening in the number of chromosomes. That is why it is called as meiosis. Thanks for watching.